I'm Abir Al-Ghamidi. I'm a PhD student at Strathclyde University, and I'm interested in studying the photophysics of protein aggregation leading to neurodegenerative diseases. This presentation will focus on the specific protein aggregation leading to Alzheimer's disease. Alzheimer's is a devastating, debilitating disease. It separates us from those around us, stealing our memories of who they are and sometimes even who we are ourselves. More than 40 million people are affected by Alzheimer's worldwide, and the numbers are expected to increase in the coming years. It was described for the first time in 1907 by the German psychiatrist Alois Alzheimer. In performing a histopathological study of the brain of his patient Auguste suffering from dementia, he brought to light the presence of two types of lesions in the brain, senile plaques and neurofibrillary tangles. Today, we strikingly can offer patients like Auguste no more help than Alois was able to 114 years ago. The situation might seem hopeless, but researchers are making great leaps in our understanding of this life-changing condition. We now believe that a key to understanding and treating Alzheimer's could lie with beta amyloid, which is the main component of senile plaques observed in the patient's brain. So how are senile plaques formed? On the neuron cell's membrane, there's this molecule called amyloid precursor protein, with one end inside the cell and the other outside the cell. It is thought that amyloid precursor protein helps the neuron grow and repair itself after injury. And like all other proteins, amyloid precursor protein gets used, and over time it gets broken down and recycled. Commonly, amyloid precursor protein gets chopped up by two enzymes, creating a fragment called beta amyloid. The beta amyloid is then cleared in the body. In the case of Alzheimer's disease, there is an imbalance. The beta amyloid is no longer regulated and is found in too great a quantity. And because beta amyloid fragments are chemically sticky, they start to aggregate with each other, forming larger structure called oligomers. These oligomers are toxic. They start to interfere with the neuron function and damage cell walls, ultimately causing cells to die. As more fragments are added, oligomers increase in size and become insoluble, eventually forming beta amyloid plaques, which are hallmarks of Alzheimer's disease. At Strathclyde, we are looking at a method that can both enable early detection of protein aggregation and also indicate the extent of aggregation over time. This technique could have significant implications in both preventing the disease and developing therapeutic strategies for sufferers. Our method focuses on an amino acid found in beta amyloid called tyrosine, which can be used as an intrinsic fluorescent probe. Intrinsic probes are unique because they allow us to detect the slightest changes in the surroundings of the probe without disturbing the native structure by using extrinsic probes. Now, fluorescent means that when light of a specific wavelength is shined on tyrosine beta amyloid, it gets absorbed instantly and the molecule becomes in the excited state. The molecule remains in the excited state for a very short while, say a few nanoseconds. It loses part of the energy it absorbed during this time. Then it releases the remaining energy it absorbed by emitting light of a longer wavelength. This process is called fluorescence, and the average time the molecule remains in the excited state is called fluorescence lifetime. Fluorescence lifetime is an intrinsic property of the fluorophore. It can be used to distinguish analytes that have similar emissions. It can also help detect changes in the fluorophore's surroundings. The lifetime of tyrosine and beta-amyloid peptides is highly sensitive to the changes in the environment induced by peptide oligomerization. Therefore, we believe that by studying tyrosine's fluorescent properties over time, we can detect changes and provide early warning of aggregation. Alzheimer's is one of the biggest medical and social challenges of our generation. It is estimated that by 2050, the number of Alzheimer's patients will grow to 130 million people. It is today the most expensive disease. The total estimated worldwide cost of dementia was around 800 billion for the year 2015. Thus, we need to invest more time and money into Alzheimer's research. It is no longer an option, it is a necessity, and we are doing relatively little to address it.